Hello and welcome to Lyra Nara. And today we have prepared a very interesting subject for you, and that is the different stages of cancer. We're going to start with the triggers, we're going to show you the hormonal changes, the acidic waste accumulation, the microparasitical infection, and all of the other things that happen in your body pre-cancer and while you're developing the cancer. And after this video, I hope that you will gain a much better understanding of what is really going on within your system with cancer, and of course what supplementation you should take and what you should pay attention to in order not to develop cancer in the first place and then deal with it once you suffer from this condition. So without further ado, let's get started and learn more about the different stages of cancer. And we're going to start, of course, with cancer phase 1. And that is the phase of the psycho-emotional trauma, because cancer is triggered approximately 1 to 3 years prior to the diagnosis of cancer. And this is when the individual with cancer experiences a traumatic psycho-emotional event that is inescapable or is on track with repeating patterns of a psycho-emotional trauma that generates so-called toxic emotions. And these toxic emotions affect, of course, the sleep patterns, and this is due to the depletion of the melatonin secretion within your body. And while most people can cope with stress very well, there are some that are more susceptible to cancer and to developing this because of this stress situation. There are those people that seem to be highly vulnerable to life stress and trauma, and they feel unable to cope when life throws a curveball their way. And these people are normally perfectionists and they live in a fear of conflict or fear of stress and trauma and loss, and they're also normally deeply frightened of negative events in their lives. And so when they are faced with a major trauma, this cancer personality, so to speak, feels trapped and unable to escape from the memory of the traumatic experience and the painful feelings of anger, hate, resentment, and grief associated with this experience. And melatonin is a very important hormone that regulates the so-called circadian rhythm and your sleep patterns. And recently it was determined that melatonin inhibits cancer growth through the ongoing production of interleukin IL-2. Interleukin-2 controls the white blood cell activity and prevents microbial infections thereby. And melatonin is the primary hormone responsible for regulating the immune system. And when there is not enough melatonin, therefore, in your body, the production of interleukin-1 and interleukin-2 is diminished. And interleukin-1 protects against infection, and interleukin-2 regulates the activities of the white blood cells, including the T cells, the B cells, the neutrophils, the macrophages, and the natural killer cells that are responsible for the immunity within your body. And when there is insufficient interleukin-2, stress-induced viral bacterial yeast infections and the bacteria, the yeast and the viruses, they're all free to invade the normal cells that damage the DNA through the release of mycotoxins within the cell nucleus. And the American Cancer Society stated, quote, melatonin has been shown to slow or stop the growth of several types of cancer cells when studied in the laboratory, end quote. The National Cancer Institute Office of CAM states, quote, both melatonin and chronotherapy have been studied for many years, but despite largely positive findings, we have not been brought into mainstream cancer therapy, end quote. Dr. Hammer discovered that every cancer has a different and very specific psycho-emotional trigger settled in a specific part of the brain and causing a brain edema there. And each brain relay center controls and is connected to a different specific organ. Dr. Hammer said in the 1980s, quote, there is at present a movement to divide medicine into organic medicine and psychological medicine or psychotherapy. When a doctor states that there is no organic cause, he is giving the psychotherapist a free hand to treat a health issue as psychosomatic condition. Such division is absurd for a good doctor because illness cannot be parceled out. The psyche, the brain, and organ are three levels of the same organism, and the course of events on them is always synchronous. End quote. So here you have then the list with the common cancers and the psycho-emotional conflicts that cause them, that trigger them in the first place. So problems with the adrenal cortex stem from a conflict of wrong direction or gone astray. Bladder problems from an ugly conflict and dirty tricks. Bone cancer from a lack of self-worth and inferiority feeling. Brain tumors from stubbornness, refusing to change old patterns, and mental frustration. Breast milk gland problems involve care and disharmony conflicts. The breast milk duct, the problems there stem from a separation conflict. The left breast is a conflict that concerns the child, the home, or the mother. The right breast is a conflict with partner or others. Bronchioles, a territorial conflict. Cervix stems from severe frustration. Colon, ugly, indigestible conflict, etc., etc. So if you're interested in all of those, pause the presentation right here and take a closer look. And with that, we come to the second phase of cancer. And during this second phase, there is an elevation in the stress hormone called cortisol. And this induces an adrenaline or epinephrine depletion. And there are unfortunately only limited reserves of adrenaline in the body, and when an individual is under this constant psycho-emotional stress, these reserves are quickly exhausted. And while insulin is used to transport glucose into the cells, adrenaline is critical for mobilizing glycogen out of the liver to supply the necessary glucose to overcome the unexpected stress situation. And thereby adrenaline is also responsible for cellular mitochondrial respiration and for converting glucose into ATP energy. And healthy cell division occurs via the metabolic pathway known as oxidative phosphorylation and via the KREBS citric acid cycle of the mitochondria. Adrenaline that stimulates the so-called G protein and the GDP molecule is an essential player in the mitochondrial cell respiration, and the glucose conversion according to the KREBS citric acid cycle and the oxidative phosphorylation metabolic pathway. And without this GDP, the cells are forced to ferment glucose. And through the fermentation, a low amount of ATP energy is being created via the process known as glycolysis, which creates lactic acid in the cell and an acidic environment, therefore. And in 1931 already, Otto Warburg was awarded the Nobel Prize for Physiology for discovering this mechanism of the mitochondrial cell respiration. And this process of fermenting glucose occurs in the so-called cytosol, which is the intracellular fluid of the cell, and causes a discharge of lactic acid there. And this discharge of lactic acid within the cell creates a low pH value with a highly acidic environment. Otto Warburg postulated, quote, the loss of cellular mitochondrial respiration due to adrenaline depletion and respectively oxygen arrest is the cause of cellular mutation toward cancer cells, end quote. And cancer cells originate from normal body cells in two phases. The first phase is the irreversible injuring of cellular respiration, and this phase is followed by a process of cancer formation, a long struggle of existence by the injured cells to maintain their structure. A part of the mitochondria perish from this lack of energy, while another part succeeds in replacing the irretrievably lost respiration energy by fermentation energy. And in fermentation, the energy is at only 2 ATP instead of the 36 ATP that a normal healthy cell has. And therefore, cancer patients are usually very low in energy and feel really exhausted. And this depletion of adrenaline is really bad news, because with the increase of fermentation, the development of cancer cells takes place gradually, and there must be a transitional phase between normal body cells and fully formed cancer cells. And when the fermentation is well developed but not so great that the respiratory defect has been fully compensated, we may have cells which indeed look like cancer cells but are clinically not cancer cells. And such cells have lately been found in prostate, lungs, kidneys and stomachs of elderly persons. And such cells have been referred to as sleeping cancer cells. Dr. W. Frieda studied in details the adrenaline depletion, and she treated this by administration of so-called dextrorotatory lactic acid to buffer the levorotatory lactic acid resulted from fermentation. Like this, the body's pH at 7.4, which is the ideal value, can be restored, and the patient's health improves right away. Let's talk also a little bit more in depth about this mitochondria respiration disruption that happens. Because a cancer patient always suffers from over-acidification of the tissues. In order to deprive the tumor of a favorable environment to proliferate, the tissue pH value must be changed from acidic to alkaline. And this is easier said than done because all alkaline foods lose their intended effect as soon as they enter the bloodstream, and they are used up in the blood for buffering before it can reach the tissue. And the body always endeavors via appropriate regulating mechanisms to maintain the blood pH value at around 7.4, which is essential, of course, for the stability of hormones, and in particular adrenaline hormone. And if the blood pH value drops, the tissue pH value rises and vice versa. And this means it should be possible to indirectly raise an unhealthy acidic tissue pH by lowering the slightly alkaline blood pH value. Over-acidification of tissue is prevented in a healthy organism by the dextrorotatory lactic acid that is constantly produced by exercising and suitable nutrition. And this indicates that an input of optically dextrorotatory lactic acid would solve the problem. This seems like a contradiction to the layman in that tissue is to be deacidified by administering an acid. The paradox disappears, however, if all interrelations are kept in mind. Acidification of the blood by means of dextrorotatory lactic acid lowers the blood pH value until both blood and tissue pH reach the same level. And this happens in cancer patients who are administered an appropriate dose of this dextrorotatory lactic acid for precisely 5 weeks. And during the period from the first until approximately the 4th day in week 6, the acid substances will be discharged from the tissue into the blood, and the pH value of which will drop for a short time to very low values therefore. And the excretion of the pathological substances of the tissue via blood, liver, kidneys and skin during this period is apparent from an entirely pungent and acidic smell of the patient. Feeling generally unwell, the patient is also irritable, aggressive and depressed at the same time. And at the height of this changeover reaction that usually lasts for about 3 days, the pH value of the tissue and blood reach the same level. We also need to talk in a little bit more detail about the so-called KREBS cycle that we've already mentioned. So the continued supply of this dextrorotatory lactic acid finally ensures an unproblematic and physiological restitution and maintenance of a blood pH value at 7.4 and a tissue pH value above that figure. And this will remove a critical precondition for continued growth of a tumor in a cancer patient, namely the acid environment. The kidneys and the liver are now capable of carrying out their full detoxification functions and thereby laying the foundations for safe removal of subsequently occurring disintegration products of a malignant tumor. And finally, the dextrorotatory lactic acid also causes the biological neutralization of the toxic laboratory lactic acid of the tumor into a non-toxic neutral form. And this is of utmost importance as it removes the stimulus for an increase in the cell division rate. Normalizing the acid-alkali balance also stimulates adrenaline production and improves its effectiveness, which is an equally important precondition for a healthy aerobic metabolism. And the therapy here introduced offers relatively great advantages to patients because the changeover reaction over 3 days, except this reaction, they are not subjected to any stress. It is neither painful nor does it cause vomiting, loss of appetite, bleeding of the bladder, or other similar side effects that are well known from aggressive cancer therapies. But of course, as with any therapy, talk to your trusted healthcare practitioner and don't do this by yourself. The dextrorotatory lactic acid is also a main ingredient in whey, which is the liquid component of cottage cheese, and that is combined with flaxseed oil to form the so-called Johanna Budwig cancer diet. And this diet has saved many cancer patients' lives and is promoted today as a so-called ketogenic diet. So you can do this as well in order to help the production of this lactic acid along. Also, stop eating too many carbohydrates and too much starchy food. Also, a glycolytic environment is associated with an anti-apoptotic and pro-proliferative state characterizing most solid tumors. So an increase of pyruvate into the mitochondria by either DCA or inhibition of LDH, promotes the glucose oxidation, increases apoptosis, and brings about a decreased proliferation and tumor growth. So what comes next? Well, next is the so-called third phase of cancer. And during this third phase, somatids, which are tiny microorganisms that are necessary for life and they live naturally in our bodies, pleomorphize into yeast-like fungus to ferment excess of glucose and lactic acid that was produced in the cells. And in a healthy person, these somatids are limited to 3 stages in their life cycle: the somatids, the spores, and the double spores. However, in a highly acidic, low pH environment created by this lactic acid, the somatids' biological cycle changes into a 16 stages cycle. And these stages include the parasitical bacterial yeast forms, which ferment the excess of glucose and lactic acid in the cell. And these pathogenic forms then migrate into the cell nucleus to reproduce, and there they release acidic waste that inhibits cell DNA repair and all-important tumor suppressor genes. And with the tumor suppressor genes such as p53 to regulate cell death, the apoptosis, when the cell has mutated beyond repair, the cell lives on and cell growth regulating proto-oncogenes turn into oncogenes and cause normal cells to mutate into cancer cells thereby. And scientists such as Gaston Naessens studied and named these minuscule microorganisms somatids. Other scientists in the 1960s, such as Dr. Alphonse Weber, considered these microorganisms to be microparasites of the type Plasmodium or CA protozoa, interfering directly with the stem cells and DNA, as he found. And we consider this latest hypothesis to be better documented and more plausible than the viral infection hypothesis or other quite mainstream ones that you hear a lot. And the trigger of all chronic diseases, including cancer, is a shocking, unbearable psycho-emotional event, as we've mentioned, and this long-term stress depletes the melatonin and the adrenaline, as we've seen, which further creates a deep distortion of the metabolic vital Krebs cycle that leads to a fermentation with excessive lactic acid invasion into your tissue. And this acidic milieu favorizes the awakening of the dormant somatids or the CA protozoa, which undergo different stages, finally penetrating and destroying the DNA and the stem cells. It's also interesting to learn a little bit more about this somatid cycle. Professor Nessens found that in healthy cells, the somatids living in the tissue, the blood, and the intracellular cytosol of the cell are limited to 3 development stages, as we've mentioned already: the somatid stage, the spore stage, and the double spore stage. And as we've learned also already, when the cell environment becomes acidic, these tiny microorganisms pleomorphize into 16 vital bacterial yeast-like fungus stages to help ferment the glucose excess and the lactic acid that has resulted from this fermentation process. And Professor Nessens said, quote, "A somatid is a basic living particle. It is indispensable to life. Without it, cellular division cannot take place." End quote. And somatids are polymorphic, meaning that they can grow from one another, from a virus to a bacterium to a fungus. We were able to grow it in a culture, and that's where we observed its polymorphisms in 2 cycles, Professor Nessens said. And first, there is a microcycle during which the reproductive hormone that permits cellular division is deployed or developed. And this healthy 3-stage cycle is stopped by inhibitors in the blood during certain illnesses, including degenerative diseases such as cancer. And this microcycle in 3 stages keeps going and becomes a 16-stage cycle during this illness. And little by little, he said, I was able to observe somatids and I extracted them from the blood. I was able to isolate it and grow it in a culture, and that's how I established its polymorphism. End quote. Let's also have a closer look at the protozoa cycle. Because the microparasites of cancer, these protozoa types discovered by Dr. Alphonse Weber, penetrate the red blood cells, the so-called erythrocytes, where they proliferate and multiply. And then they break and dissolve the capillary blood vessel walls using the so-called lysosome enzymes. And at this stage, the parasites invasively migrate extravascularly into your connective tissue matrix and the organ's parenchyma, where the organ's tissue is destroyed, thereby creating what we know today to be a degenerative chronic disease such as cancer. And the final stage of this CA protozoa infection is marked by the invasion of the stem cells. And the first defense mechanism of your stem cells is so-called vacuolization. So they enclose the parasites in small blisters within them. They capture them, they make prisoners, so to speak. And when there are too many vacuoles, the stem cell starts dividing in order to minimize the number of vacuoles that are stuffed with the parasites within it. And this is known as mitosis and leads to hyperplasia. And once the nutrients for the parasites kept enclosed in the stem cells vacuoles have dried up, the stem cells start dividing in order to reduce the number of parasites per cell, or there is a stem cell apoptosis, so cell death, due to the toxicity and the starvation of the stem cell. And at this stage, the microparasites are freed again, and depending on the organs that are invaded and the general patient's health state, the first symptoms appear in form of skin rashes, skin eczemas, joint and lymphatic system inflammations and edemas, as well as dysfunctional up to total shutdown of organs functionality. And a cancer tumor is the latest stage of this infectious process that develops in 4 stages. First, we have the so-called protozoa intravascular compensated infection, which is in your blood vessels, when the protozoa are contained within your blood vessels. The second stage is the protozoa intravascular decompensated infection, also within your blood vessels. Then the third stage is the extravascular compensated infection, where the PROTOZOA migrates from your blood vessels into your connective tissue and into your organs. And the fourth and last stage is the PROTOZOA extravascular decompensated infection, where the PROTOZOA migrates into your stem cells. And the cancer stages through CA PROTOZOA infection are very similar to malaria, toxoplasmosis infection, also developments of the PROTOZOA class. The next phase would be phase 4. And during phase 4, the lack of adrenaline causes a depletion of dopamine in your brain. And adrenaline is made of dopamine, and as more and more dopamine is used up during the stress, the amino acid tryptophan creates serotonin to offset this depressed mood. And this subsequently results in a depletion of tryptophan, which is needed to synthesize niacin and niacinamide, which is vitamin B3, that is vital for cellular respiration. And niacin is converted by tryptophan into NAD coenzymes, which are subsequently used by the KREBS citric acid cycle in the mitochondria for glucose conversion, cell respiration, and ATP energy, of course. And without tryptophan and niacin, the KREBS citric acid cycle and the oxidative phosphorylation metabolic pathway is broken. In the 1950s, Dr. Abraham Hoffer, Director of Research at the Regina Psychiatric Services branch in Canada, Saskatchewan, accidentally found a treatment of vitamin C and niacin while administering high doses of oral vitamin C to patients that were hospitalized with psychiatric disorders, of course. And Hoffer effectuated a cure in a number of psychiatric patients who had incurable cancer. Dr. Hoffer subsequently treated cancer patients in his own private practice from 1967 to 2005 using vitamin C and niacin with a life extension from 5 to 5.7 months to 100 months on average. And in cancer patients, we see a chronic niacin deficiency caused by a chronic tryptophan deficiency. And it takes about 60 milligrams of tryptophan for the liver to synthesize 1 milligram of niacin in the body. Adrenaline, as we've mentioned, is made by dopamine, and the more and more the dopamine is used up during the stress, the amino acid tryptophan synthesizes serotonin to offset this depressed mood. And this subsequently results in a depletion of serotonin and tryptophan, meaning that the niacin cannot be created or converted by tryptophan into NAD coenzymes for this very crucial KREBS citric acid cycle for healthy cell functioning. And this depletion of NAD or NAD is a big problem. All the tissues in the body convert this absorbed niacin into its main metabolically active form, and that is NAD, or nicotinamide adenine dinucleotide or cleotide. More than 400 enzymes require this NAD to catalyze reactions within your body, which is more than for any other vitamin-derived coenzyme. NAD is also converted into another active form, the coenzyme nicotinamide adenine dinucleotide phosphate, or NADP for short. In all tissues, it is produced except the skeletal muscle. And if this critical glucose-converting ATP energy-producing factory is shut down, then the cell is of course forced to ferment, rising glucose levels via the process known as glycolysis to obtain the reduced amount of ATP energy that it can still get. And with that we come to the fifth phase, and during phase 5 the deficiency in adrenaline causes also ascorbic acid, better known as vitamin C depletion, in your adrenal glands. And ascorbic acid is the main ingredient that is used by dopamine to make noradrenaline, which is then converted into adrenaline. And during prolonged chronic stress, more and more adrenaline is pumped out and then depleted, meaning more and more ascorbic acid is used up in the regeneration of adrenaline. And during chronic stress, the adrenal glands also release ascorbic acid into the body to diminish the stressful impact of adrenaline on your heart and on your blood pressure systems. And ascorbic acid is essential for preventing cell DNA damage caused by oxidative stress, but also for converting oxygen waste products, the superoxide and the hydrogen peroxide, into oxygen and water within the cell mitochondria during this oxidative phosphorylation that we've already discussed. And the loss of ascorbic acid thereby increases the cell mitochondrial DNA damage and induces cell mutation. Ascorbic acid is an essential nutrient required by the human body for survival. It is a key antioxidant used to break down superoxide and hydrogen peroxide, as we said, in the mitochondria, but also to prevent cell DNA damage. And it's an essential ingredient required by the adrenal glands to produce stress hormones, including norepinephrine and adrenaline. And during this prolonged chronic stress states, ascorbic acid is used to create norepinephrine or noradrenaline to be converted into epinephrine or adrenaline. And the dopamine uses the ascorbic acid and the oxygen to synthesize norepinephrine via the pathway known as dopamine beta hydroxylase. And for this reason, there is 100 times more ascorbic acid in the adrenal glands than anywhere else in your body. And during prolonged chronic stress, ascorbic acid is also released from the adrenal glands to protect the heart from cardiovascular disorders and arrhythmias. Yet, as the condition of prolonged chronic stress continues for months and years and adrenaline levels are continually depleted, vitamin C levels are also depleted, as both are inextricably linked with each other. And without an adequate supply of ascorbic acid, superoxide and hydrogen peroxide, you are unable to break down this into oxygen and water within the cell's mitochondria, as we've already discussed. And normal healthy cells are more likely to mutate, therefore, into cancer cells due to the cell DNA damage. So what has this depletion in vitamin C have to do with tumor growth? Well, during normal cell respiration within the mitochondria, the superoxide and the hydrogen peroxide are damaging byproducts of this oxidative phosphorylation that must be converted into oxygen and water in order to avoid further cellular damage. Ascorbic acid and the enzyme is called superoxide dismutase are the key substances used to convert these harmful substances into oxygen and water and thereby avoiding cell mutations. And this is the antioxidant effect of ascorbic acid that occurs within normal cells in order to prevent cancer. Yet the body has ingeniously designed ascorbic acid to have the opposite effect inside of cancer cells, where instead of it being used as an antioxidant to remove superoxide and hydrogen peroxide, it is used as a pro-oxidant to create superoxide and hydrogen peroxide to cause cell death within the cancer or the tumor cell. And in a landmark study conducted by the National Cancer Institute and the National Institutes of Health in the USA, researchers documented this pro-oxidant mechanism of ascorbic acid within tumor cell in mice models and found high levels of intravenous ascorbic acid, also vitamin C, and significantly reduced tumor cell growth. And the results were that given the relative sensitivity, the efficacy of pharmacologic ascorbate administration on the growth of OVCAR5, PAN02 and 9L tumors was examined in nude mice. Treatment commenced after tumors reached a palpable size of 5 to 7 millimeters in diameter. Xenograft experiments showed that parenteral ascorbate as the only treatment significantly decreased both tumor growth and weight by 41 to 53% for OVCAR5, so this is a type of ovarian cancer, PAN-O2, which is a type of pancreas cancer, and 9L, which is a glioblastoma. Metastases present in about 30% of 9L glioblastoma controls were absent in ascorbate-treated animals, and those were the precise quotes from the results of this study. But does vitamin C literally kill cancer? Well, this preclinical data that provides a firm basis for advancing pharmacologic ascorbate in cancer treatment to humans is great. The tumor xenograph results are especially noteworthy because ascorbates of vitamin C was considered a nutrient, was used here only as a single agent drug in this test. So the only drug that was administered was vitamin C. So we have clear results for the effects of only this particular ingredient. Pharmacologic concentrations of ascorbate decreased tumor volumes, as we've mentioned, up to 53% in diverse cancer types that I've shown you some of, known for both their aggressive growth and the limited treatment options that they have. And the findings showed, quote, that pharmacologic ascorbic acid concentrations were cytotoxic to many types of cancer cells in vitro and significantly impeded tumor progression in vivo without toxicity to normal tissues. The amelioration of ascorbate cytotoxicity in vitro by the addition of catalase was consistent among sensitive cancer cells and points unambiguously to hydrogen peroxide generation in the extracellular medium. Hydrogen peroxide cytotoxicity is promiscuous in its action, compromising membranes, glucose metabolism, and DNA integrity. Variation in the sensitivity among cancer cells may be related to the complex networks that hydrogen peroxide acts on, combined with the range of functional mutations intrinsic to each cancer cell line, which are not present in normal cells. And although the molecular basis for the relative resistance of normal cells remains to be elucidated, the current in vivo data supports that pharmacologic ascorbates, so vitamin C concentrations, which can readily be achieved in humans, diminished growth of several aggressive cancer types in mice without causing apparent adverse effects. And although our preclinical mouse data showed that tumor growth was significantly decreased, the use of pharmacologic ascorbate as a single agent was not curative. As modalities in cancer are often combined, this data suggests that pharmacologic ascorbate in combination with other therapies deserves further exploration and for treatment of cancers that otherwise have poor outcomes, such as pancreatic and ovarian carcinomas and glioblastoma. And the proposed mechanism for tumor-killing actions of pharmacological ascorbate are that ascorbate distributes from the blood to the tumor extracellular fluid compartments after IV administration. And within the tumor cell, ascorbate is then oxidized to ascorbate radicals by metalloprotein catalyst, which donates an electron to the oxygen, forming thereby superoxide radicals, and ultimately the tumor-killing effector hydrogen peroxide. And vitamin C was first suggested to have cancer-fighting properties in the 1930s already and has been the subject of controversy ever since, of course. And despite repeated reports of selective cancer cell toxicity induced by high doses of vitamin C treatments in vitro and in mouse models, the mechanism of action has remained elusive to this day. And with that, we come to what we call the sixth or final stage of cancer, where the immune system has shut down because of your subconscious want or will to die that you might not be aware of, and also because of this elevated stress hormone cortisol production that leads to the levels of serotonin and dopamine depleting in the brain, and that causes, of course, then depression as well. And an individual experiencing this inescapable shock and prolonged chronic stress often feels tired of life and deep down wants out of this never-ending struggle of pain and life, and he sends thereby the subliminal messages to the immune system, and the immune system responds. And this occurs at the subconscious level, where the immune system receives orders to stop production of interleukin-2, producing T cells and B cells and natural killer cells, as well as macrophages and neutrophils. And while at the conscious level, the individual with cancer usually has a strong desire and will to live, it is at this deeper subconscious feeling level that the individual with cancer feels overwhelmed by the pain and the struggle of life, and sending thereby out subliminal thoughts to this immune system of his or hers to shut down and exit life and finish this situation that is very unpleasant. And without immune system cells, the viral, bacterial, yeast-like fungus that have pleomorphized within the cells continue to grow, and the newly created cancer cells continue to multiply. And this death wish can be very subtle, and often individuals have mixed feelings and do not even realize that they have this death wish in the subconscious mind. And this awareness indicates that this can be often caused by a rejection of one who was loved, and this can be caused by the loss of a loved one, by a person feeling unloved or unwanted or rejected. And in this kind of attitude, the cancerous condition is truly unstoppable. You really have to take care of your state of mind when you deal with any chronic disease. Your state of mind and feeling during the cancer therapy will make or break the therapy, will help you heal yourself or will help you die quicker. The real commander-in-chief is you. And this awareness indicates that basically the real final cause of cancer is related to your subconscious death wish, and you must get rid of this at any price in order to make any type of therapy work. These cancer conditions are prevalent in most human beings, but the bodies of those who wish to continue living, who have enthusiasm for life, who are loving and feel loved, these bodies are generally too healthy and too strong. The immune system is too well developed and works too well for cancerous conditions to gain control of their cells. Remember that if you don't like the fact that the mechanism to take you to your exit has kicked in, connect to the opposite side, which is life. Remember that you have not exited yet, and for as long as there is a life within you, you are life itself. So live. And with all that being said, I hope that you've enjoyed this presentation. If you did so, please do not forget to like, share and subscribe. If you have any questions about holistic treatments for cancer and what is going on in your body, do not hesitate to contact us, shop@lyranara.com. I'm also going to post the complete contact information, including our toll-free phone numbers, in the description box below for you. And until next time, stay healthy.